Good morning. Welcome to the series of knowledge where we can learn about photosynthesis. I am Mara Flor Tinatagkan. What makes plants special? What plants do? Plants don't really eat in the normal sense of the word, apart from venomous fly traps. Plants can make their own food. Unlike animals, they consume food to eat to make energy. As it turns out, plants are getting their energy directly from the source of sun. Plants use sunlight as well as water and carbon dioxide, which are plentiful in our earthly surroundings, to produce their own nourishment through a cellular process called photosynthesis. What is photosynthesis? It is the process used by plants to make their own food. It is conversion of light energy into chemical energy. Photosynthesis makes sense because photo comes from the Greek word for light and synthesis means to build. So plants use light and small molecules to build a bigger molecule that they can then metabolize to produce energy. So let us learn about how photosynthesis works. Photosynthesis consists of two stages. There are so-called light reactions in which solar energy is converted into chemical energy. And then there is a Calvin cycle which uses the chemical energy to build P3P and the precursor to glucose and other molecules. Both of these stages occurs inside one of the organelles found in the plant cells, which are called chloroplasts, and there are around 30 to 40 of them per cell. Let us focus first on the light reaction. The energy absorbed from the chlorophyll during the light reaction is used to power photosynthesis system 2 that breaks the bonds of water absorbed through the plant's roots. Third oxygen atoms bind with each other to form the gas O2. O2 is byproduct of photosynthesis not used by the plant, so it is released through the stomata of the plant. Stomata are little pores in leaves that open and close to let oxygen out and carbon dioxide in. When water molecules break apart, the remaining two hydrogen atoms have a positive charge and are called protons. These protons are kept inside the thylakoid by the thylakoid membrane. When there are more protons inside the thylakoid than in the stroma outside, protons want to leave the crowded thylakoid. When the protons cross the membrane to leave, a proton uses their passage to power ATP production. The light energy absorbed in chlorophyll also powers photosystem 1 that charge up the energy carries molecules NADP plus into NADPH. NADPH then carries its energy over the power and dark reactions or Calvin cycle. To summarize the light reactions, protons are absorbed by the pigment to power photosystem 1 and photosystem 2. Photosystem 2 split water molecules into two protons and oxygen atoms are expelled as O2 gas through the stomata. Protons cross the thylakoid membrane and power proton complex ATP synthase to make ATP. NADP plus is powered by the photosystem 1 to make NADPH to be used in the dark reaction. So the light-independent reaction finished with charged NADPH, ATP, and release O2. Next is the dark reaction, also called Calvin cycle. The dark reaction starts and ends with the same product, hence cycle. All dark reactions take place in stroma of the chloroplast. CO2 is reduced by the reducing power generated in the first step and carbohydrates are produced. Carbon fixation reactions produce sugar in the leaves of plants from where it is exported to other tissues of the plant, a source of both organic molecules and energy for growth and metabolism. The end product of light reactions, ATP, NADPH, are used to fix CO2, occur both in the presence or absence of light. The carbon dioxide fixation reductions into carbohydrates can occur via three pathways. The first one is Calvin-Vincent cycle, reductive pentose pathway. Fixation and reduction of one molecule of CO2 requires three molecules of ATP and two NADPH. It is occurs in the mesophyll cell chloroplast, which is the CO2 acceptor is RAB-P. The RAB carboxylase enzyme is needed also to produce the stable product of 3-PGA. So the second one is the C4 or Hatch-Lock pathway. First product is 4-carbon oxaloacetic acid. These are the steps for this pathway. First is carboxylation of the step to OAA. Step carboxylase is enzymes involved. Second is the reduction of OAA to malate or aspartate. Then the decarboxylation of malate in the double sheet cells to form pyruvic acid. The transfer of pyruvic acid to the mesophyll cell, which is the fixation of carbon dioxide to form the 3-PGA. This is the process of Kranz anatomy. So the Kranz anatomy is specialized structures of C4 plants where the mesophyll cells are clustered around bundle sheath cells in the ring-like fashion. The last one is the CAM. CAM or crassulacean acid metabolism pathway. It operates in orchids, pineapple, and other succulent plants wherein the stomates are closed during the day and open during the night. Thank you for watching.